வணக்கம். Welcome to NCERT Biology Reading, Class 11, Chapter 13, Photosynthesis in Higher Plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their food needs. The green plants make or rather synthesize the food they need through photosynthesis and are therefore called autotrophs. You have already learnt that the autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotrophs. Green plants carry out photosynthesis, a physicochemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living forms on Earth depends on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. It is the primary source of all food on Earth. It is also responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of photosynthetic machinery and the various reactions that transform light energy into chemical energy. 13.1 What do we know? Let us try to find what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, green pigment of the leaf, light, and carbon dioxide are required for the photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was particularly covered with black paper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of light. Another experiment you may have carried out where a part of a leaf is enclosed in a test tube containing some potassium hydroxide cotton which absorbs carbon dioxide while the other half is exposed to air. The setup is then placed in a light for some time. On testing for the presence of starch later in the two parts of the leaf, you must have found that the exposed part of the leaf tested positive for starch while in the portion that was in the tube tested negative. This showed that carbon dioxide was required for the photosynthesis. Can you explain how this conclusion could be drawn? 13.2 Early experiments. It is interesting to learn about those simple experiments that led to gradual development in our understanding of photosynthesis. Joseph Priestley, 1733 to 1804, in 1770, performed a series of experiments that revealed the essential role of an air in growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that candle burning in a closed space, a bell jar, soon gets extinguished. Figure 13.1 A, B, C, and D. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air, but when he placed a mint plant in the small bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove. Can you imagine how Priestley would have concluded the experiment using a candle and a plant? Remember, he would need to rekindle candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup at the one used by Priestley, but by placing it once in the dark and once in the sunlight. Jan Ingenhousz, 1799, showed that the sunlight is essential to the plant process that somehow purifies the air by fouled by burning candles or breathing animals. Ingenhousz is an elegant experiment within aquatic plants showed that the bright sunlight small bubbles were formed around the green parts while in the dark they did not. Later he identified these bubbles to be of oxygen, hence he showed that it is only the green part of the plants that could release oxygen. It was not until about 1854 that Julius von Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. His later studies showed that the green substance in plants, chlorophyll as well as we know it now, is located in special bodies later called chloroplasts. Within the plant cells, he found that the green parts in plants is where glucose is made and that the glucose is usually stored as starch. Now consider the interesting experiments done by T. W. Engelmann 1843-1909 using a prism. He split light into a spectral components and the illuminated a green alga cladophora placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolution. He observed that the bacteria accumulated mainly in the region or blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption of spectra of chlorophyll A and B discussed in section 13.4. By the middle of the 19th century, the key feature of the plant photosynthesis were known, namely the plant could use light energy to make carbohydrates from carbon dioxide to water. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was understood as carbon dioxide plus H2O in presence of light, which will give CH2O plus O2, where CH2O represented a carbohydrate, example glucose, a 6-carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by microbiologist Cornelius von Neil 1897 to 1985, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrate. This can be expressed by 2H2A plus carbon dioxide in presence of light gives 2A plus 2A plus CH2O plus H2O. In green plants, H2O is the hydrogen donor and is oxidized to oxygen, O2. Some organisms do not release oxygen during photosynthesis. When H2S instead is the hydrogen donor for purple and green sulfur bacteria, the oxidation procedure the product is sulfur or sulfate depending on the organism and not oxygen. Hence, he inferred that the oxygen evolved by the green plant comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore 6 carbon dioxide plus 12 H2O in presence of light, C6H12O6 plus 6H2O plus 6O2, where C6H12O6 represents glucose. The O2 released is from water. This was proved using radioisotope techniques. Not that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given above? 13.3, where does photosynthesis takes place? You would of course answer if the green leaf are in the chloroplasts, based on what you earlier read in chapter 8. You are definitely right. Photosynthesis does takes place in the green leaves of plants, but it does so also in other green parts of the plants. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually, the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplasts will be aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is a membranous system consisting of grana, the stroma lamellae, and the matrix stroma. Figure 13.2, there is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH in stroma. Enzymatic reaction synthesis sugar, which in turn forms starch. The former set of reactions, since they are directly light driven, are called light reactions. Photochemical reactions, the latter are not directly light driven but are dependent on the products of light reactions ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reaction carbon reactions. However, this should not be construed to mean that they occur in darkness or in that they are not light dependent. 13.4, how many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves? Even in the same plant, we can look for an answer to this question by trying to separate the leaf pigments of any green plant through paper chromatography. A chromatographic separation of the leaf pigments shows that color that we see in the leaves is not due to a single pigment but due to four pigments: chlorophyll A, bright or blue-green in the chromatograms, chlorophyll B, yellow-green, xanthophylls, yellow, and carotenoids, yellow to yellow-orange. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of chlorophyll A alpha pigment to absorb light to different wavelengths. Figure 13.3a. Of course, you are familiar with the wavelength of the visible spectrum of the light as well as the VIBGR. From figure 13.8, can you determine the wavelength color of light at which chlorophyll shows maximum absorption? Does it show another absorption peak at any other wavelengths too? If yes, which one? Now look at figure 13.3b showing the wavelengths at the maximum photosynthesis occur in a plant. Can you see that wavelengths at which there is maximum absorption by chlorophyll A, that is in the blue and the red regions, also shows higher rate of photosynthesis. Hence we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 13.3c, can you say that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? These graphs together show that most of the photosynthesis takes place in the blue and red regions of the spectrum. Some photosynthesis does not take place at the other wavelengths of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophylls, and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelength incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. 13.5. What is light reaction? Light reaction or photochemical phases include light absorption, water splitting, oxygen release, and other formation of high energy chemical intermediates. ATP and NADPH, several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes, LHC. Within the photosystem 1, PS1, and photosystem 2, PS2, these are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The light harvesting LHC light harvesting complex are made up of hundreds of pigments molecules bound to proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system also called antennae. Figure 13.4. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystems. In photosystem PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called P700, while in PS2 it has absorption maxima at 680 nanometer and is called P680. 13.6. The electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by electron acceptor, which passes them to an electron transport system consisting of cytochromes. Figure 13.5. This movement of electron is downhill. In terms of an oxidation reduction or redox potential scale, the electrons are not used up as they pass through the electron transport chain, but are passed on the pigments of photosystem 1, PS1. Simultaneously, electrons in the reaction center PS1 are also excited when they received red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a great redox potential. These electrons then are moved downhill again, this time to molecule of energy rich NADP plus. The addition of these electron reduces NADP plus to NADPH plus H plus. This whole scheme of transfer of electrons starting from the PS2 uphill to the acceptor down to the electron transport down to the electron transport chain to PS1. Oxidation of excitation of electrons transfer to another acceptor. The finally downhill to NADP plus, reducing it to NADPH plus H plus is called the Z scheme. Due to its characteristic shape, figure 13.5, this shape is formed when all the carriers are placed in a sequence on a redox potential scale. 13.6.1. Splitting of water. You would then ask, how does the PS2 supply electrons continuously? The electrons that were moved from photosystem 2 must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with PS2. Water is split in 2H plus and O and electrons. This creates oxygen, one of the net product of photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. 2H2O is equal to 4, it gives 2H2O gives 4H plus O2 plus 4 electron minus. We need to emphasize here that the water splitting complex is associated with photosystem 2, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and oxygen formed likely to be released? In the lumen or on the outer side of the membrane. 13.6.2. Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. The process through which ATP synthesized by cells in mitochondria and chloroplasts is named photophosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first photosystem 2 and then the photosystem 1, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain, as seen earlier in the Z scheme. Both ATP and NADPH plus H plus are synthesized by this kind of electron flow, figure 13.5. When only photosystem 1 is functional, the electron is circulated within the photosystem and the photophosphorylation occurs due to a cyclic flow of electrons. Figure 13.6. A possible location where this could be happening in the stroma lamellae, while the membrane or lamellae of the grana have both photosystem 1 and photosystem 2, the stroma lamellae membranes lacks photosystem 2 as well as NADP reductase enzyme. The excited electron does not pass on to the NADP plus but is cycled back to the PS1 photosystem 1 complex through the electron transport chain. Figure 13.6. The cyclic flow hence results only in the synthesis of ATP but not of NADPH plus H plus. Cyclic photophosphorylation also occurs when only light of wavelengths beyond 680 nanometer are available for excitation. 13.6.3 Chemiosmotic hypothesis. Let us now try to understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism, like in respiration, in photosynthesis too. ATP synthesis is linked to development of a proton gradient across a membrane. This time these are membranes of thylakoid. There is one difference through here the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria when electrons move through the electron transport chain. Chapter 14. Let us understand what causes the proton gradient across the membrane. We need to consider again the processes that takes place during the activation of electrons and their transport to determine the steps that cause a proton gradient to develop. Figure 13.7. Since splitting of the water molecule takes place in the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoid. B. As electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of the electron, which is located towards the outer side of the membrane transfers its electron not to an electron carrier but to an H carrier, hydrogen carrier. Hence, these molecules removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side of the lumen inner side or the lumen side of the membrane. Then C, the NADP reductase enzyme is located on the stroma side of the membrane along with electrons that come from the acceptor of electrons of photosystem 1. Protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in lumen there is an accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as the measurable decrease in pH in the lumen. Why are we so interested in proton gradient? This gradient is important because of its breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the CF0 of the ATP synthase. The ATP synthase enzyme consists of two parts. One called the CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides through enough energy to cause an conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy-packed ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATP synthase. Energy is used to pump protons across a membrane to create a gradient or a high concentration of protons within the thylakoid neuron. ATP synthase has a channel that allows diffusion of protons back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. Along with the NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing carbon dioxide and synthesis of sugars. 13.7. Where are the ATP and NADP used? We'll see in next video, next part. Thank you.